I didn't realize when I finished my PhD that the date of finishing and graduating, it's very important for my next steps. You want to apply for grants, fellowships, and it all starts ticking when you graduate as a PhD. Most of the grants do have a 7-year application deadline from graduating that I was not aware of.
Welcome to EHA Unplugged, the official podcast channel of the European Hematology Association, EHA. I am your host, Isabel Olivera. Today's guest is Meritxell Alberich Jordà, a principal investigator based in Prague in the Czech Republic at the Institute of Molecular Genetics and at the Second Faculty of Medicine at Charles University in Motol Hospital. Her work focuses on hematopoietic and leukemic stem cells and how their dysregulation drives acute myeloid leukemia, AML. But this conversation isn't just about the science. It's about the journey behind it. In this episode, Meri reflects on her path to becoming a PI, sharing with us what she wishes she had known at the start of her research career. Welcome to this episode.
Do you consider yourself a hematologist? I did study biology, and very soon in my career, I got an interest in hematology, and I have always been studying in this field, but I'm not an MD, so not a real hematologist.
Where did you start? When did you become interested in hematology?
So I studied, in fact, biotechnology at the University of Barcelona. That was very theoretical at that time, and I was missing experience in the lab. And I got a chance to go abroad to the Netherlands for an Erasmus exchange, and it was supposed to be 4 months. And that, in fact, was extended to 1 year because I absolutely fell in love with the bench. But at that time, I also realized that biotechnology was not really my field. I had a big interest in the medical research. And I found a position at the Erasmus MC at the Department of Hematology, where I actually applied for a PhD, and that was already in hematology.
What was your PhD on?
So I was studying the effect of the cannabinoid receptors in blood cells, in leukocytes, how they affect myeloid differentiation. We all know cannabinoid receptors for being expressed in brain, and we know the effects that they might cause if you stimulate them. But there is another family member, another cannabinoid receptor that is expressed in blood, and that one was totally underexplored. So under the mentorship of Ruth Delwell, we explored the role of this cannabinoid receptor in hematopoietic differentiation and in leukemogenesis.
Did you continue this subject after your PhD, or what did you do after?
So during the PhD, we mostly focus on membrane receptors, G-protein coupled receptors, and how they activate signaling pathways. But very soon we got interested in understanding deeper the molecular mechanisms that get activated when we stimulate these receptors. And we moved to understanding transcription factors. And that brought us to CBP alpha, which is a key transcription factor in myeloid differentiation. So because of that, I searched for a postdoc in the field of transcriptional regulation in myeloid differentiation. So in fact, it was like a spin-off.
How did you develop as a scientist as a postdoc?
So if I have to tell you the truth, I was not very sure whether I wanted to do a PhD or even a postdoc. But things just happened. The opportunities came. And when I was finishing my PhD, I got to meet Dan Tennant, that was my mentor in Boston, and he offered me joining his lab. I wasn't sure that the postdoc was a good fit for me, but I decided to give it a try. I thought it would be interesting to move from the Netherlands to the States. That's how it went. I started on the topic of CBP alpha, transcriptional regulation in myeloid differentiation. And then as the time passed, the project evolved, and I ended up moving to another transcription factor from the same family, that was CBP gamma. And also we explored how CBP gamma can control myeloid differentiation and again plays a role in the development of acute myeloid leukemia. And the project kind of expand, evolve, brought new research lines, and I took the one that I thought was more suitable, and that was how the project evolved.
What did you want to do after that? Did you want to become a PI?
No, that was also not in my plans. In fact, I always thought I would end up working in a company. That was what I thought I could do. And when I was thinking about the transition from postdoc to my next step in my career, I did search for that opportunity. And in fact, I quit the lab thinking that I was moving to a company. But it didn't work out, in fact. I went to Prague. That's where I was moving at that time for family reasons. And I had an agreement with a company, but it didn't work out. So I ended up with no job in a country where I did not speak the language. So that was a bit challenging. Didn't want to stay at home. So I thought, okay, the only thing I can do here is, in fact, go back to the lab. That is what I know. So I had some contacts in Czech Republic, and I talked to the director of the Institute of Molecular Genetics at that time, Václav Horeši. I explained the situation. I'm jobless. And he said, well, we can offer you a job, but there is not an open position for group leader, but you can build up to you. So I went there. I asked for funding. In a short period of time, I was able to recruit some funding, set up some projects, and soon after, I got the opportunity to establish myself independently at IMG. That is where I'm still right now.
It sounds easy, but it's not, because you had to apply for funding for a project on your own line. How did you find your own line of research?
In Boston, I spent 7 years, and that gave me time to focus on my main projects. But we also had some side projects that were coming up as we were doing our work. Some of them established the basis for future research that in principle I was leaving behind. However, when the plan changed and I had to set up my own lab, I contacted my former PI, explained the situation, and I said, then I'm interested in following up those things that I left halfway or were just starting. Would you be supportive? Would you be willing to have a collaboration with me? We could do this together. And I was very lucky because then Tenan was open to support me, to giving me animal reagents and things that would really help me to establish my own projects. But again, it was as a follow-up of what I was doing. Later on, when the lab started running after 2-3 years, we finished the initial projects and we expanded a bit more towards stem cell biology, inflammation, acute inflammation, but also chronic inflammation. And that's what the lab has been working on during the last years. So we still do myeloid differentiation. We still focus on leukemia, mostly AML, but we have been also getting more interest in the immune regulation of stem cell biology.
What do you think are the main findings you associated with?
During the last years, we have been working a lot on chronic and acute inflammation and how those conditions affect stem cells, their fitness, their functionality, their fate. And I think that I'm still, I guess, part of those studies we did in the transcription factor, CPI, alpha, CB gamma. But I think that during the last years, we have evolved towards something that was more ours, more from our lab. We have developed our own models, in vivo models, to study these conditions of inflammation. And I think that those are the studies I'm the most proud of, I would guess.
If you have to choose one project?
Well, I think more than a project itself, what I really have been enjoying during these years as an independent scientist is the effect I had on my students and the mentorship I could provide them. Science is important. I love the projects we have and the research we do. I like to understand every insight of the stem cells and how they react to external cue. But overall that, I think that the most important thing is the work I have been doing with my PhD students, the interactions, the collaborations, training, mentorship, helping them to grow up as a scientist. I think that's the nice thing about my job.
Are there any of your findings that are finding an application?
So the work we have been doing on AML during the last years has been in a very close collaboration with a company, French company, Advanced Biodesign. And those are really on the course of clinical trial phase 1 with a specific compound a company developed, and we provide all the murine data to validate findings and then allowing us to move to human clinical trials for relapse and refractory AML. So that's what I would think is the most translational thing we have ever done, but it did not really stem from my own lab. It was in collaboration with a pharmaceutical company. The other side of the lab is working on really fundamental projects, really basic research. I think my work will not really directly lead to a clinical trial product or drug development, because that's not what we are focusing on. But I think that we are providing the knowledge to establish the basis for understanding how we can therapeutically target, for instance, chronic inflammation, and to prevent the effects that this condition, which is actually very prevalent in our society. I mean, think about it, autoinflammatory disorders, cancers, diabetes, many conditions do have this hallmark of chronic inflammation. So we know now, based on our studies and many others, that this chronic inflammation affects the stem cells, not only in, let's say, in the muscles or in other systems, but also in the hematopoietic system. So by preventing the effects that this chronic inflammation might have on the hematopoietic stem cells, we might be preserving the risk of developing other hematological disorders. So we are not really focusing on drug targeting, but we provide the basis to build up on that.
Can you explain how did you become involved in EHA first?
So when I was a PhD student, I already joined some of the meetings at EHA. That was really at the early days of EHA. And then when I was thinking about the step towards the postdoc, I was suggested to apply for an EHA fellowship. And I wrote a fellowship. At that time, I think we were only giving 2. So it was the early days of those grants and fellowships, but I was awarded one of them. And in fact, that helped me to go for my postdoc abroad. And after that, soon after obtaining the fellowship, I was able to also enroll in several committees. So I participated at that time in the scientific program committee, the research committee, then I jumped to the fellowship and grant committee, later on to the stakeholder committee. So I have been actually in many, many committees at the EHA through all my career. And I think that has really helped me to have this community of scientists, hematologists around me that really have been very, very supportive and inspiring. And also has helped me a lot to build up a network, which I think it's very important in the job we do, that we don't get just closed in our labs, focus only on what we do, but we also expand to really see what's happening in the clinics. What are the needs there? How can I contribute to that? And I think EHA has been part of all this interaction and development.
Yes, EHA is more associated in general with clinical research, but you are really the example that a researcher can also benefit and have an important role participating in EHA. And
I think that that's a bit maybe the old view of EHA, to have mostly an association based on clinical research and clinicians. But during the last years, we have been expanding a lot. We have been opening ourselves more towards fundamental research, physician scientists, promoting them. And I think that now the research community is also enjoying EHA. A couple of years ago, I was elected as a board member, which allowed me to bring the voice of researchers to the association. And I hope that with this little step that I do, I can also push a little bit more the association to open their eyes and opportunities for researchers to be also part of the community. And I think that it's maybe taking a while, but step by step, we are really getting there. So I'm very happy about that.
Do you think that hematology is a field where there is a particular close relationship between research and clinics, where many research findings are being translated quickly to the clinics?
Yes, but this goes in both directions. I think that we as scientists really have to pay attention to what's happening in the clinics when we are thinking about what to base our projects on, what to develop, what are the needs in the clinics. And then the research or the results that we provide can be applied or used to think about therapy. So I think it's a two-way relationship. So scientists can benefit, I think, from working closer with clinicians. And I think clinicians can also benefit when working closer with us researchers.
So what are your main activities at the moment?
So this is interesting because at the beginning, when you establish your lab, you're still very close to what's happening on the bench. I did like a lot, actually, pipetting. So as soon as I could, I would be there with a team doing experiments. As the time passes by, you get other duties. You are teaching at the faculty. You are on several committees at the university, PhD, student association committees. I'm very actively at EHA, participating at several committees, the board, the executive board. So you start being less close to the bench and more involved in what I call administration, which is not admin by itself. But yeah, right now I spend most of the time on the computer, either writing, reviewing, assessing, preparing presentations. But I keep time every week to talk to my students, to still know what's happening in the lab, what are they dealing with, what are the troubles, and try to help them to move those projects forward.
Is there one lesson that you wish you had known when you started?
There are many, actually, but there is one that I think is very important. I didn't realize when I finished my PhD that the date of finishing and graduating, it's very important for my next steps. You want to apply for grants, fellowships, and it all starts ticking when you graduate as a PhD. Most of the grants do have a 7-year application deadline from graduating. That I was not aware of. So I was enjoying my postdoc, and I spent 7 years in Boston as a postdoc without thinking that if I wanted to be a PI, which in principle was not the case, but if I wanted to be a PI, I had to keep in mind that the deadline for grant applications was 7 years from graduation. The 7 years depend a little bit. Some agencies is 3, 4, some others is 10, some are flexible. But that's something very important to keep in mind when you are on the transition from postdoc maybe towards a PI.
Is there anything else you would like to add for the audience that I haven't asked? Something that was important in your career?
I think it's important to be aware that sometimes you should not have very high expectations. You should know what you want, and that's fantastic. Go for it. But sometimes things will happen, opportunities will appear, and science is like a trip, right? And it's not only about the end goal, it's also the ride you have along. So try to enjoy each of the steps that you are going through. Sometimes they can be very tough. Opportunities will come up. You have to search for them, but do not obsess on the end goal. That's what I always tell my PhD students. They see the end goal as getting the PhD, but that takes 4 or 5 years. So enjoy also all the little steps that you gain through that process and be proud of them, not only the end goal.
Thank you very much for sharing with us your journey.
Thank you, Isabel. Thanks to you.
Thank you to the audience for listening. If you enjoyed this podcast, please like it and stay tuned for more episodes of EHA Unplugged.